Hi, my name is Natasha Hungerford, and today I'd like to tell you about stingless bee honey's unique properties as a novel source of the beneficial disaccharide cellulose. This has been a collaborative project, as you can see from a long list of names. So Australia has about 12 species of stingless bees that live in hives and which do not sting. Stingless bees are the largest group of eusocial bees. Typically, they're about 4 millimetres in length. They are compact and darkly coloured. Shown here is a comparison with the regular honeybee. Stingless bees can be domesticated in artificial hives and are often regarded as people's pets. So here's an example hive here. Stingless bee nests are made of wax and resin and are typically built in natural crevices. The common stingless bee in southeastern Queensland is Tetragonula carbonaria, and the brood comb of this species features a spiral. The other common species is Tetragonula hawkinsi. Stingless bees have a distribution to tropical and subtropical parts of the world, including tropical areas of America, Africa, Southeast Asia, and Northern Australia. There's over 500 species of stingless bees that have been described, and with many yet to be described. So we wanted to look at the honey of the stingless bees, so we had access to honey from 5 species, 1 from Brazil, 2 from Malaysia, and 2 from Australia. So stingless bee honey has long been prized by Indigenous communities in the tropics, providing an ancient source of sweetness with health benefits. Traditional Indigenous uses such as treating wounds, burns, or respiratory illnesses. In Australia, stingless bee honey is called sugar bag honey, and it was prized by Aboriginals who collected it from wild hives. Stingless bees store their flavoursome honey in clusters of small resin pots, such as depicted here, on the extremities of the nest. Each hive produces only small amounts of sugar bag honey, less than 1 kilogram per year, so it's a special product to be savoured and relished and valuable to the bees as food for themselves and their larvae. In warmer climates, the hives can produce excess honey. We wanted to characterise stingless bee honey, and the first step was analysis of the sugar content of the honey. So what we found was that stingless bee honey contained 3 sugars: fructose, glucose, and an unknown disaccharides. So the common disaccharides include lactose, sucrose, and maltose, and it wasn't any of these. And it didn't match any of the disaccharide standards we had in our possession. Previously, when people had looked at stingless bee honey, they thought that this disaccharide, they thought it was maltose, but had never been separated or characterised. So we set about subjecting each stingless bee honey to purification methods by HPLC and collected this large peak shown in red here. After freeze drying the resultant solution, we obtained this white powder, and then we used advanced techniques such as mass spectrometry and NMR to characterise the disaccharide, and we identified that it was in fact trihallulose. So trihallulose is an isomer of sucrose with an unusual glucose-fructose linkage, and I'll describe more of that later. So we were then able to buy the standard of trihallulose. It's a pretty rare sugar and it's very expensive to buy the standard. And we found by looking at the spectra and comparing them, so we're pattern matching here, in effect, we found that the standard trihallulose matched exactly the trihallulose we had isolated from the honey. We then looked at the chromatograms on our LCMS, and we found that the standard indeed on this system also matched the trihallulose that we'd isolated from the honey. So you can see the standards in blue, the standards in red, the one we isolated from the honey is in blue, and you can see that they line up and that they're distinct from other sugars that we had in our collection, platinose in green and sucrose in black. And then if we look at all 5 of the honeys, indeed the peak lines up. So trihallulose was present in all 5 species from those 3 different countries. So this was a very interesting find. So this was the first report of trihallulose naturally present at a significant level in a food. So this disaccharide had been discovered previously in the excrement of whiteflies and had been shown to be highly beneficial in having both a low glycemic index and a low insulinemic index. So low glycemic index means that it's more slowly digested and doesn't cause a spike in blood glucose after it is consumed. So that's important for diabetic control. Trihallulose is also known to be akaryogenic. So unlike sucrose, trihallulose is less easily metabolized by microbes that cause dental caries. So i.e. trihallulose doesn't cause tooth decay. And it's also an active antioxidant. So these properties attributable to trihallulose may be responsible for the reported beneficial health properties of stingless bee honey. And this work generated a lot of interest when it was published last year. So we then collected 36 honey from 4 different species from Australia and Malaysia. So the top 2 species are the Australian species and the bottom 2 are the Malaysian species. And we analysed the sugar content. So across the bottom here, this is sugars present in grams per 100 grams. And you can see from the orange bars that there's a significant amount of trihallulose in each of the honeys. You can see that there's no sucrose shown in green and there's no maltose detected in any of the honeys, with glucose and fructose also present and a tiny amount of a trisaccharide, which we identified in the same way that we identified trihallulose, and we found that that trisaccharide was in fact urlose, so another unusual sugar. So if we look at the averages by species of the sugars in the honey, you can see that Geniotrigona thoracica, this Malaysian species, has the highest amount of trihallulose, and that is certainly significant in each of the other honeys as well. So we wanted to find out the origin of trihallulose in stingless bee honey. So we had some hypotheses. First of all, though, if you look at sucrose, it's made up of a glucose portion shown in blue and a fructose portion shown in grey. And trihallulose has these same monosaccharides, glucose and fructose, joined together. The only difference is they're joined in a different way. So sucrose is joined at 1 to 2 and trihallulose is joined 1 to 1. So we thought that the bees could either collect trihallulose directly from natural resources in the environment, and potentially the resins that they use to make the pots that they store the honey in could have some factor that could cause a conversion to trihallulose. The other option is that trihallulose is synthesized from nectar sugars within the stingless bee's gut. So nectar sugars typically are sucrose, glucose, or fructose in varying amounts. So this is a possibility. So it either happens in the bees themselves, or the transformation could also happen in the honey. So we wanted to do some feeding experiments, and we made some microcolonies of the Australian species Tetragonula carbonaria and allowed the bees to settle in their new home by allowing them to forage free for 3 days. And then we confined them for 24 hours with access to only sugar solutions instead of nectar. So we then could look through this perspex window to see which pots were filled, and those were the honey samples that we collected. So here's a schematic of our experiment here. So the feeder was either contained a sucrose solution with water or a glucose fructose solution in water. And we're then able to collect the honey from the pots that were filled in the period of confinement, and we obtained what we called lab honey. You can see that it was quite different looking to natural honey, which is much darker in colour. We're then able to use advanced analytical techniques to analyse the honey that resulted from these feeding experiments. And what we found was that sucrose was converted completely to trihallulose, mostly, a larger amount of urlose than is observed in natural honey, and fructose in grey down the bottom here. Only a little bit of glucose was formed, presumably because the glucose is incorporated into the urlose, this structure here. So this was a remarkable discovery that sucrose is converted by the bees to trehalulose. And we did the next experiment then, which was feeding glucose and fructose separately to the bees, but the honey we obtained in this case was largely unchanged. So it seemed likely that the bees can't convert separate fructose and glucose into trehalulose, and this small amount observed here we thought was due to contamination from before the feeding experiment. So these experiments have confirmed that trehalulose is produced in the gut of the bees from sucrose, and we conclude that the likely source of trehalulose in the honey is sucrose in nectar. So to get our honey with optimal amounts of trehalulose, we need to find a nectar with high sucrose. We then wanted to find out whether sucrose could be transformed to trehalulose in the honey. So we did some storage experiments with 2 species of bee, 3 different temperatures, and 2 treatments. So pasteurized. So pasteurization is a technique used by many beekeepers to prolong the storage of their honey, and they typically do this at 63 degrees for 30 minutes. And we also did an unpasteurized treatment for comparison. And you can see here, this was very fresh honey, so there was still a little bit of sucrose at time 0. But in all cases, for all temperatures, the sucrose was converted to trehalulose, particularly for Hocking's eye, and as a little bit maybe to some of the monosaccharides in the Carbonara species. So comparing pasteurized and unpasteurized treatments, there was no significant difference between the groups. So this suggests that the conversion of sucrose to trehalulose is unlikely due to bacteria. So this is just a bit of a summary then. Our sucrose is converted in the honey, whether it's pasteurized or unpasteurized. It doesn't come from glucose and fructose alone. It doesn't come from the environment, and the bees convert it into trehalulose. And we need nectar high in sucrose to get honey high in trehalulose. So the sweetness of trehalulose is approximately 60% that of sucrose. Potential for trehalulose to be adopted as a natural sweetener alternative to sucrose with health benefits. Trehalulose has potential as an authenticity marker for this unique honey. So stingless bee honey sells for around $200 per kilogram, which is up there with the price of Manuka and royal jelly honey. So a high commercial value makes it at risk for substitution, where people could sell other honey as stingless bee honey or dilute the product. But this research, we can test for this novel sugar, and therefore trehalulose can act as an authenticity marker. So that's great for the industry. People have patented ways of making trehalulose synthetically with enzymes and bacteria, but our research shows stingless bee honey can be used as a whole food on its own or in other food to get these same health benefits. So we found that feeding bees sugar syrups can result in trehalulose formation, but this gives a fake honey, which is frankly a worry. So it is a sum of all of the components of honey which make it such a valuable product. So the honey we produced in the lab was a fake honey. Are we able to distinguish this from natural honey? And the answer is yes, by another lab technique, isotopic testing, and we can tell the difference between honey that's produced naturally and those from sugar feeding. Of course, it's not good for bees' health to be only fed sugar solutions, so this wouldn't be an option for beekeepers. Honey contains a complex range of phytochemicals, all important for brood rearing and colony expansion. And keeping of native stingless bees is gaining popularity in Australia for their role as pollinators and for their unique honey. There is undoubtedly a lot more to find out about stingless bee honey. Thank you, and we thank these organisations for their support.